Hi, my name is Casey Day, and I'm going to be presenting some of the simulation modeling work that my lab has done recently in support of the management of fish populations in Washington State. So by way of a brief outline, I'm going to give an overview of the software we developed, which is CDMetaPOP, and sort of our approach to simulation modeling. And then I'm going to briefly present three case studies on three different species that occur in the same study system, and those are west slope cutthroat trout, bull trout, and the non-native brook trout. So when I think about simulation modeling in support of animal populations, I like to think of simulations as sort of as a problem-solving tool more than perfect replication of reality or some kind of magic 8-ball, but really a way to explore problems, identify processes and mechanisms and key variables within a model, and explore sort of alternative scenarios. There are a number of ways that you can utilize simulation models that can be sort of grouped into three general categories of questions. So there are theoretical questions you can ask with models. There are analytical or methodological questions you can ask to basically to test if your methods work. And then there are empirical applications where you can build a simulation model of a real-world system, explore alternative management scenarios, alternative decisions, and those are the kinds of applications that I'm going to be presenting today. So in terms of a general framework in approaching the role of simulation modeling in riverscape genomics, on a riverscape, there are a lot of processes going on simultaneously. There are environmental processes such as land use change and climate change going on. And then there are the genetic processes, population genetics and evolutionary processes. Sort of in the middle of all that are the managers trying to balance all these different factors and variables going on in their system and try to make the best decisions that they can in support of conservation and management of populations. And so we can think of these in terms of riverscape connectivity on the environmental side and how that affects genetic connectivity. And riverscape genomics provides this nice framework to bring all these things together. And where I think about simulation modeling fitting in here is I like to think of it as sort of providing a foundation or a framework that the managers can fall back on to, explore questions, explore problems, look at alternative management scenarios when they really don't have the ability to do all these things in the field, and they can fall back on a simulation model. A recent review of fish models said that because salmon populations show watershed scale variation in demographic and genetic traits, we propose that models combining demographic, genetic, and spatial data are promising tools for improving their management and conservation. So that's the type of simulation model that we've developed in our lab, and we've called it CDMetaPOP. And at its core, it is a metapopulation model where you've got multiple subpopulations or patches, and you can have migration and dispersal between patches. But then in our model, we populate those patches with individual animals. That's where it differs some from a traditional metapopulation model. And we can track individual movement, individual growth, and we also track individual genotypes. Then we can measure the movement of genes across the landscape. Then on top of this sort of network of populations, we add these spatially explicit layers, and these can be things like environmental factors like temperature that may affect survival or may affect growth. Could be harvest or angling or anything like that. And so the model is both spatially explicit and individual based, but it's in sort of this metapopulation framework. So within CDMetaPOP, there are a lot of processes going on. Some of those can be controlled by the class or the size of the individual. Some can be controlled by the patch or the environment. Some could be controlled by both, like mortality, for example, could be age related, but also could be related to environment. And we're tracking all these parameters for each individual in the system, up to hundreds of thousands of individuals and at least thousands of patches. And we're tracking, as I said, individual genotypes as well. So the study system that I'm going to talk about today is here in northeastern Washington. It's the Lower Pend Oreille River, and it's occupied by west slope cutthroat trout, brook trout, and rainbow trout. Used to be occupied by bull trout, but they've been extirpated. And here's the Lower Pend Oreille River watershed, and then there are three sub-watersheds within there. some of our models only address the Sullivan Creek and some the larger study area. Recently, this system had a relicensing for a large dam in the area, and as part of that relicensing, there's been some mitigation efforts going on, things like bull trout reintroduction, brook trout, non-native brook trout suppression, fish passage at dams, dam removal like this dam at Mill Pond, and habitat restoration. So a lot of our work has been related to these relicensing goals. So the first case study I wanted to talk about was hybridization between the native West Slope cutthroat trout and rainbow trout. And these two species can hybridize where they overlap, but they have differences in thermal tolerances. So some prefer warm water, some prefer cold water. So we wanted to look at how a gene that acts as a driver for spatial selection through a genotype environment association related to temperature could affect likelihood of hybridization between the two species, and then also how that might interact with the presence or absence of barriers on the landscape. And I'm just going to show one figure from this paper. We've got on the y-axis here, we've got simulations that had barriers in place and some that did not have barriers in place, and then some simulations that had the selection mechanism in place and those that didn't. And I want to focus your attention here on the bottom right, and the green line specifically shows the increase in hybrids over time relative to West Slope cutthroat trout and rainbow trout. And you can see that when barriers are in place and the selection mechanism is also in place, that hybrids do the worst in those scenarios. And so we can use this information to present to the managers to think about how barriers might be promoting conservation and are not always such a negative thing in preventing movement, that maybe they're mitigating unwanted hybridization between species in some cases. So the next case study is bull trout reintroduction. This has not taken place yet, but we wanted to explore some alternative scenarios associated with the potential reintroduction of bull trout. So in terms of management scenarios, we looked at alternative ways to source individuals for the reintroduction. So you could source from populations with very high genetic diversity, low genetic diversity, from many populations, from few populations. We wanted to see how that might affect genetic outcomes like genetic diversity and genetic structure on the back end. And then again, the presence and absence of barriers. And then we also looked at some life history things like dispersal rates. We looked at low and high carrying capacity, which is basically unknown in the system, right? So again, just one figure from this paper. But one thing I wanted to point out here is that if you look where these scenarios have these overlapping circles, essentially what you've got on the y-axis is an F statistic of genetic structure, and on the x-axis you have time. And the different shades of blue and the different shades of red represent different scenarios for sourcing the reintroduction. And what we're seeing is that no matter how you source the reintroduction from many populations or few populations, that after a couple hundred years, you just don't get much differentiation among scenarios, which was a surprising result for us. The only case where that didn't really hold true was when we had a low carrying capacity scenario with barriers in place, and we had very unpredictable demographics and a lot of populations going extinct, in which case you have a lot of variation in genetic structure at the end of the simulations. So again, something for managers to think about is how to source their populations. And then finally, this is a simulation model of brook trout, which are non-native and unwanted. And we wanted to simulate the eradication of brook trout through electrofishing suppression and through the use of Trojan Y chromosomes. And this Trojan Y chromosome program involves developing males in a hatchery that have 2 Y chromosomes. Then you release those fish into the wild, and all of their offspring are male. So you skew the sex ratio in the population towards male and away from female, and you continue doing these releases, and eventually the females are eradicated and the whole population is wiped out. So we wanted to create a simulation model of this and help managers think about what kinds of potential outcomes are possible and where they might want to focus their energy in terms of monitoring. So these are the results from a sort of generic scenario, the most likely management scenario. And here you can see is where we start suppression, the population declines, and then we start outplanting YY males, and you get this differential in male versus females, and the sex ratio begins to be skewed. And eventually, after 25 to 30 years, the population goes extinct. And then we also looked at occupancy over time, so not just total population size, but this is occupancy of individual patches, and you get this inverse sigmoidal sort of shape. And it turns out that some patches have a harder time being eradicated than other patches. And we looked at this in terms of the landscape, and those patches that are more sort of centrally located or more connected within the landscape take longer to be eradicated. So that's something the managers can think about or focus on when they're trying to remove brook trout from the system. Then we also wanted to look at the fitness of the YY males and how that might affect eradication. Fitness is really unknown, and so we looked at competitive fitness or survival, and then also reproductive fitness. And what we found is this purple color here. This purple color here indicates longer time to eradication. What we found is there's this threshold around a competitive fitness value of 0.6, below which simulations basically failed to be eradicated after 100 years of treatment. And it seems that the simulations were much more sensitive to this competitive fitness value than they were to reproductive fitness. So again, somewhere managers may want to focus more on YY survival than on their reproductive fitness. So in closing, just a few things. As I said before, simulation modeling is a great way to force you to think about your system and question the assumptions that you're making about your system. Every time I've built a system model, I always find myself correcting the assumptions that I have been making in order to calibrate and build a model. The whole process of simulation modeling has really helped foster a great collaboration with the management agencies that we work with, and we have this sort of back and forth adaptive management where we present model results, and then we can compare those to their empirical data, and then we can use those data to update the model, provide new results. And it's this cycle, and it's really helped us develop a great relationship with these management agencies. And then a little about CDMetaPOP. I just want to mention that it's a really flexible tool. It can do aquatic as well as terrestrial systems. Really any system where you can describe the demographics and the movement of a population, you can run simulations and you can track genes across the landscape. If you're interested in CDMetaPOP, you can download it at GitHub, and there are plenty of publications out there to look at as well. So thank you so much for your time. I was really glad to be part of this symposium.